Very good morning, participants. Welcome to the yet another session, new batch of June, the 3 days workshop of molecular biology. Everyone, you're most welcome to the EduFabrica under the flagship of EduFabrica Molecular Biology Workshop. Good morning, everyone. Good morning, Chandrama. Good morning, Johnson. Good morning, Shweta. Good morning, Suman, Tanusri, Anjum. Everyone, a very good morning. So we wait. So what we usually do, I send you the link 5 minutes before the workshop, and we wait for yet another 5 to 7 minutes, and then we start our session around 11:10 or 11:08. So by that time, everyone will join the session, and we can initiate the lectures with everyone being in the uh, in the this YouTube streaming. So let's wait for another 2 or 3 minutes, then we start. ਮੈਨੂੰ ਆ ਬੋਤਲਾਂ ਭਰ ਦੋ ਪਾਣੀ ਠੰਡੇ ਦੀਆਂ ਤੇ ਚਾਹ ਕਾਫੀ ਨਾਲ ਨਾ ਦੁੱਧ ਨਾਲ ਚਾਹ ਤੇ ਮੈਨੂੰ ਗਰਮ ਚੀਜ਼ਾਂ ਅਵੋਇਡ ਕਰ ਕਾਫੀਆਂ ਚੱਲ ਠੀਕ ਹੈ Okay, everyone, I think has joined the session today. Good. So, our first 20 minutes or 15 minutes, we give our time for introductory part. I will speak something about myself. We will discuss a little bit about the objectives, what we will discussing in these 3 days workshop, and get to know about yourself, from where you all, from which university, where you studying, in which year you are, which course are you doing, what are your hobbies and everything. And yeah, so let me start with my thing. Oh, Abhijit, thank you for the coffee. I really need this at the time. Okay. So whom I? So basically, I had my bachelor's education um, from Singh College at the Punjab Technical University in Punjab, and then I worked for like 5 months as a lecturer in Singh College of Engineering and Technology from January to May for 5 months. And then from there, my urge to learn more didn't stop there. So I went to Germany at the Justus Liebig University in August 2010. And then I did my work in the cancer field. I didn't had my master thesis in the field of agriculture, but I switched to the human medicine, yeah, and the identifications of novel endothelial specific molecules involved in the regulations of tumor cell extravasation. And then after finishing this, I went for the molecular medicine for my PhD in 2013 at the University of Göttingen, at the city of Naturwissenschaftler, the scientists. Then I got my chance to do my project on redox regulation of PP1 in cardiomyocytes. So overall, after finishing my studies, I came back to India as assistant professor, Mata Gujri College. From August 2018 till April, I worked for like around 9 months. And then I joined in 2020 as online professor with EduFabrica, where I was teaching various subjects abroad, MBBT, biochemistry, proteomics. And nowadays, we are running these small workshops of 3 days of molecular biology, enzyme engineering, vaccine technology, and yeah, medicinal biochemistry. Beyond that, I was also AP at Rama University in Kanpur for like around 8 months. Now I'm dedicated to EduFabrica for the moment, and let's see where the future goes. Yeah, so that's something about me. and the objectives for 2 days workshop or 3 days workshop, 2 hours each day. We will meet us at the same time from 11 a.m. till 1 a.m. Yeah, so we'll always meet from 11 a.m. to 1 a.m. So the nucleic acids, so we will discuss in these on the day one all about nucleic acids, the genetic materials, type of DNA, DNA replications, model of DNA replications, DNA repair system. Yeah, then we'll discuss. Yeah, we will discuss basically everything related to your molecular biology, the basics of molecular biology, transcription, translation. Um, yeah, gene expression. So this content will be covered in within upcoming 2 days. More than that we will cover, actually more than this. Just a second, students, I need to call my seniors in order to clarify this with my medicinal biochemistry. uh, uh, Hello, hello, good morning, मैम. मैम कैसे हैं मैम? बढ़िया. बस मैम वो क्लास चल रही थी अभी, तो मैम वो बच्चों का मैसेज आया था मैम मेडिसिनल बायोकेमिस्ट्री का. आ, वो ना लाइक जो वैक्सीन का है वो अराउंड मैम वन वन है मैम. 1 से 3 है, क्योंकि कोई यशा महमूद स्टूडेंट था, शी वॉज सेइंग दैट को 2 से 4 है. ओके, ठीक है मैम. अराउंड 3 और 3:30, 3:15. ठीक. ठीक. तो 15-20 मिनट के बाद कर देंगे उनको ज्वाइन. ठीक. इसे मैं कर देता हूँ. थैंक यू मैम, थैंक यू. Just a second, students. Good. So I have discussed so far with you about myself, uh, about the objectives, and we will share our classes. Our time is fixed now. I think there is no clashes because there's lots of classes is going on during this weekend, and we need to manage each student effectively. So this class will be on YouTube mostly from 11 to 1 p.m., 11 a.m. to 1 p.m. In this afternoon session for Friday, Saturday, and Sunday. And the medium of communication would be English, because with my previous experience, the some students they can't speak Hindi. थोड़ा सा डिफ़िकल्ट होता है समझना. तो हम जो कॉमन जो लैंग्वेज है सबकी, दैट एवरीवन कुड अंडरस्टैंड, वी कीप दैट लैंग्वेज एज इन इंग्लिश. Okay, and we will whatever the things that we are talking at the moment, it was recorded on this YouTube channel. So I have created a playlist within my YouTube channel named as uh, um, Molecular Biology Workshop June Batch. So I had a workshop in the May batch also. So you are the second session is going on. So this will be your second batch. So you can see these recorded videos being there. And there are also another platforms like your Zoom, Google Meet. And the problem with them is that when we are teaching, if somebody goes out because of the network issue or something, and we don't get to know about if somebody wants to get in again. But in this platform, that's the beauty of this platform. You can join in anytime, and because of some issue you want to go, you can go out and join it again. So I don't have to control that thing of admitting the students. Yeah, and moreover, the recorded session will be on this YouTube cloud, so you can watch it anytime, anywhere, for your own benefits. So by seeing many benefits, I'm taking these classes. And second thing is, if you have any query, if you have any questions about anything, any field, you can write me in the chat box. I will try my answer. I will try my best to answer them. Yeah. So so medium of communication is English. The source of giving the online lectures will be YouTube. Yeah, you can participate, you can converse with me over the online chatting. Yes. If in sometimes what happened, it happened just now actually in the morning. I shared with you the link. There was a low band length network. There was a network problem, so the YouTube streaming doesn't started. So I shared it again. So this could happen. If this happens, please text me on the WhatsApp or just call me. I will try to solve that too. And we have discussed just now the objectives and the medium of these presentations. These 3-day workshop is actually the basics of molecular biology. We will not do any advanced things here. We will understand about the structure of DNA, RNA, transcription, translation, nucleic acids. Moreover, we will go a little bit about Lacoperon system. These systems we will understand a little bit. We will discuss about a little bit about DNA repair system, right? Further, various types of DNA, A DNA, B DNA, and Z DNA. We will discuss about them too. So all these things will be shared in upcoming days. Okay. Um, so please do subscribe to this channel if you want to have more workshops like this in future. Or I'm also sharing some good stuff related to for your, if somebody is interested to go abroad, or somebody interested to how to make sharp resume. So I'm trying to make some small YouTube videos in order to help students to carry out their dream fulfill. So I will share those links later upcoming days. So, and also if you want to interest to for on the long term to be in touch with me, join me over the LinkedIn. A connection is always good. To being connected with my students, I always love them, to know what they are, where are they going, where they are getting up. It's always good to know. So my name is Simranjit Singh, comma ph.d.d. You can join me there anytime. So now it's your turn, students. Tell me about yourself, that is your name, your class, your course, year, place you belong, like your university. Yeah. And your hobbies, and where do you see in upcoming 5 years. So I will get to know that, are you undergraduate, postgraduate, MBBS, forensic science, zoology, genetics, microbiology, or PhD student, because I have a mixture of students always coming up in these workshops. So it's now your turn, students. Tell me about yourself. Hi, Johnson. Okay, let's start. Abhijit Birdar, um, first year MBBS student, studying Paravera Institute of Medical Science, Ahmednagar, Maharashtra. Yeah, most welcome, our futuristic medical doctor Abhijit. Dr. Abhijit, you're most welcome. So these courses that you're doing, you might be studying them in during your course studies in MBBS, but not in details. You will be more into the diagnosis and diseases part in the medical. But if you're interested for the MBBS student, I'm saying, uh, but after some time, if you get into a really good college or university, and they have also research perspective there, so it's after some time I'm seeing from the doctor's point of view, medical doctor's point of view, it's get very lame to just check patients, diagnose them and get back to your bench. Yeah, you're seeing patients, this will be your everyday job, and you will be doing it for a whole year. Of course, it's a very noble cause to do, but I'm saying from the psychology perspective of as a doctor, because then you will solve many complex cases, you will save many lives, and that's the doctor's work is. But if you continue R&D also, this will keep burning a desire to know more, to do something more what has been done so far and what we can do within that field, whether it's to create a new technique to heal one cancer, or to get a new technique to get a better operation in the orthopedic level, or anything. So that's good. From the first year only, MBBS students are joining these kinds of courses of allied sciences. So the combination of these, believe me, I have seen many excellent professors from the medicine field, MD, PhD. They had really, yeah, very good tenure and very good positions in the upcoming time. So all the best. Johnson Daniel MK. I'm doing my second year BSc Biotechnology in Spicer University, Pune. Most welcome, Johnson. Most welcome. So I mean, the BSc students who are doing 3 years BSc, they also have very good perspective for the upcoming time. So you have only 1 year left. Either you go for MSc, this will be very beneficial, or you go for MBA also you could go for, or to foreign university for your masters anywhere in the world. So everything is open for you. You have to decide what you have to think about it, because after bachelor's you might get a job of 25 or 30,000 package per month, but on the long run, in order to grow within this life sciences, you need at least post graduation. So good morning, sir. Arvind Gupta, Samundra Anand. Some from where? Samprananand, medical biochemistry postgraduate. Most welcome, Sumande, professional medical laboratory technologist, University in West Bengal, Health University, hobbies reading. Yeah, Sumande, I have many people joined in actually from as a lab technologist. So today I got a request actually, Madan Singh. So this LinkedIn is amazing actually. This is amazing. So this guy, he did his um masters in clinical microbiology, then joined in 2000 after his master. Initially, he was a lab technician for 1.5 years in the hospital. Then he's a lab technician, and then he joined as a in Balaji Action Medical College. Then after he did his masters, he got as a Dr. Lal Path Lab. He worked for 4.5 years there. Then he's a senior scientific officer at the Max Healthcare. So things gradually increases within this field also. So this was good to know that um, that how we grow within this field. Okay, Anjum era BSc MLT. I joined to know how it will happen in the lockdown to get any job. Anjum, you should have joined me with the MBBT course. We are giving a very nice certificate with the molecular biology and biochemistry technique. So that might be have been more beneficial to you from the job perspective, because that course that I have created for 1 month, which is still going on, I started on 6th 10th 11th of June actually. It's been only 1 week. We have just discussed the basics, so I think I will also discuss the basic here of the same here too, and from Monday onwards we will continue with the technical part there. So that will be really beneficial to add into your CV, that you had a 1 month course. We will also do internship in that. We will write about the review article, many things. So it has many benefits from the job perspective. So many of our students actually, they told me that so because of that certificate, they were preferred over the jobs. Because even I was the assistant professor at the college, everything was fine, getting good salary package, and suddenly they said they want to lock down. They don't have money. They want to do cost cutting, and all university go out. All sorry, faculty go out. So we were like 8 faculties in the university, and out of eight, five were kicked out. So this happened. Tanushri Biswas. Hello, sir. Myself, Tanushri Biswas. I've completed my postdoc in biotechnology from University of Burdwan in 2019. Okay, most welcome, Tanushri. Most welcome. Abhijit Bradar, after 5 years, I will start preparation for NEET postgraduate. I didn't get you, Abhijit. Jigmate Lahaza, internship student of traditional medicinal medicine. After 5 years, acha, okay. After 5 years, what you wanna do? Okay, okay. Acha, you will help students who wants to prepare for NEET. Okay, yeah, that's also a good thing actually. It's a very big thing, market for that. Oh, Jigmate Lahaza. Okay, Chandrima Ghosh, most welcome, Jigmate. I hope your dreams comes true. Chandrima Ghosh, MSc Microbiology from JI University, interested to go abroad for PhD, job purpose in biotech, biopharma industry. Good, good. This is Abhi, completed my PG in biotechnology, currently working at Rajiv Gandhi Center for Aquaculture. Oh, wow. Avihan, I want to be an assistant professor in medical college. My best wishes for you. After my bachelor's, I would like to go for abroad for my higher studies. Yes, that's possible. Yes, sir. I had a specialization on biophysical chemistry, so I want to work on this field if I get a chance. Oh, biophysical chemistry. That's good. Biswajit Swain. I'm currently first year MS in biochemistry at the Ravancha University, Cuttack, Odisha. Want to see myself research field for good TI. Okay. So I have one. Most of the questions they are saying that they want to go abroad for their PhD or masters. And I have these questions many times. There are many times. Is this link not working? Okay, first let me share. Um, Dr. Shweta Mishra. So students, I have shared this link, Europe calling. It's a small 10 minutes video, 13 minutes video. Because I was sharing, everyone was asking me this again and again and again and again. So I just shared this, just go through that. You will get your answer in that, most of the questions. If you have any other questions, feel free to ask me also later. Okay, so that was very nice introductory session for all of us. Um, yeah, so let's start with the lecture now. So most likely our lectures will be for 40 minutes. Then we will take 5 to 8 minutes break afterwards, because the retention period of one student is around 40 minutes only. One person could study with the concentration with 40 minutes. Then person should take 5 to 6 minutes break and then start again. So we'll follow this rule here in our workshops also. I will not continue straightforwardly one on one. I will stop it and will come to your screen. If you have any questions, I will be happy to answer that. Yeah. Okay. Let's start the session then. Internship will be you have to write in review article. I can tell you later, Anjum. You can contact me, so I can express everything about that later. So let's start. First, we discuss a little bit about structure of DNA. So I'm pretty much sure you all had cleared your class 12th with good grades, and you might be all aware of basics of molecular basis of inheritance, which you have as a class 12th lecture. Just a second, I'm coming, student. So, structure of DNA. So in this part, we will discuss mainly the concept of DNA, its various structure, packaging into the nucleus of the cell, and to demonstrate the concept of central dogma of life, how the central dogma of life works out. So nucleic acid, if we take the history, it was initially founded by Friedrich Miescher in 1869. So he is the first one who have took the initiative in the 18th century and show us a pathway of nucleic acids by the Friedrich Miescher. And then Frederick Griffith, he did the transformation experiments on the bacteria in order to find out that what was the reason behind the transformation of DNA, like from which part the genetic material is being transferred, whether it's DNA, protein, or RNA. Then Rosalind Franklin, he got very fine X-ray photography of DNA in 1952. Yeah, Rosalind Franklin. So it was a very nice X-ray photograph in which they were showing this double helix structure of DNA. But he was not able to convince people what things are what is nucleic acids, what are nitrogenous base pairs. He was not aware of that. But then Watson and Crick, they came in 1953 and they describe the DNA molecule from the Franklin X-rays picture. So they discovered and they explain each part: hydrogen bonds, pyrimidines, purines, your glycosidic bond, phosphodiester bonds, double helix structures. So everything was explained in very well manner. Then the Chargaff rule claims, which says that there's equal amount of purines and pyrimidines are present in the in your DNA. So this, this, this. So this, this, like this. So it's a anti-parallel to each other. So DNA consists of, we know that here is a sugar and phosphate backbone with a nitrogenous base pair inside attached with the help of hydrogen bonds here. So adenine and guanine, they belongs to purines, and thymine and cytosine, they belongs to pyrimidines. So the purines will have equal amount of pyrimidines always. This is a Chargaff rule. And adenine and thymine, they will make a double disulfide bond, oh sorry, double hydrogen bond, and guanine and cytosine, they make a triple hydrogen bond. Cool. So molecule of DNA is made up of millions of tiny subunits called nucleotides, and there is a prokaryotes, that is nucleoid and plasmids, and eukaryotes are chromatin of nucleus. And these DNA structures, they have these nucleotides which are made up of nitrogenous base pair, pentose sugar, and phosphate group. First is your, in DNA, it's deoxyribose sugar, in which on the second prime carbon atom your hydroxyl group is removed, and another side OH group is present. So if we start from 1, 2, 3, 4, and there will be another carbon here, 5. So 5 carbons are there. So this is on the 5th carbon your phosphate group will attached, and yeah, this is deoxyribose sugar, and this is your nitrogenous base pair. So these are two kinds of sugars, that is ribose sugar and deoxyribose sugar. Ribose is present in the RNA, and this is present in the DNA. OH group, H group, like this. This is OCH2, OCH2, yeah. And the bases are, as we discussed, purines and pyrimidines. The purines includes your adenine and guanine, and these are membered of 5 and 6 membered rings together. And whereas the pyrimidines, there is a thymine and cytosine, they are only 6 membered rings. That's the basic difference between your purines and pyrimidines. And together, these purines and pyrimidines, they are attached to the sugar group like this, as we have discussed earlier. And then these sugar groups are attached to the phosphate group. So here is the sugar phosphate ladder on the background, on the side chain, and in the center is your various nitrogenous base pairs. And to each nitrogenous base pair, we will have another ladder on the other side corresponding. And remember one more thing, that the bond between your sugar and phosphate, this bond is called as phosphodiester bond. This is phosphodiester bond, and this bond is known as the bond between your nitrogenous base pair and sugar is your glycosidic bond. This will be the glycosidic bond, and this one will be your phosphate bond, and so on. So there's a difference between your nucleosides and nucleotides. Basically, nucleosides are the one without phosphate. They have only sugar and base group. They mainly consist of sugar and base group, nitrogenous base group and deoxyribose sugar. So there are 4 kinds of nitrogenous base pairs we have, that is your adenine, guanine, cytosine, and thymine. Yeah. So deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. So these will be known as your nucleosides. But into the same, if we attach the phosphoric group, your phosphate group is added. So we have to just add in the same name monophosphate at the end. Deoxyadenosine monophosphate, deoxyguanosine monophosphate. So they will be abbreviated as DMP, DGMP, DCMP, and DTMP, and so on. Yeah. So that's the basics of your nucleosides and nucleotides. So this is typical, as we have discussed, nucleotide structure. So these nucleotide structure joined together by the phosphodiester linkage. And the two purines and pyrimidines, they are paired with the help of hydrogen bonding. So adenine paired with thymine and cytosine paired with the guanine, and they're all paired with the help of hydrogen bonding. And this always, whatever the amount of pyrimidines are, it will be the same amount of purines will be there. If we having this sequence called A, T, C, G, T, C, A. So what will be the corresponding sequence in the DNA then, students? Tell me. If this is the sequence, tell me in the chat box what will be the corresponding sequence in the ATCGTCA. Type me in the chat box what will be the corresponding sequence of ATCGTCA. I hope you have got my question. My streaming goes like 2 minutes late maybe. So just let me know in the chat box what will be the corresponding sequence to ATCGTCA in the chat box, बच्चे. TAGCAGT. That's right. Not ACTGCTA, Suman. It's the basic. We go for corresponding. T corresponding, A corresponding with the A with the T, G with the C, C with the G, A with the T, G with the C, and T with the T. Yeah. So like this sequence will go. Okay, good. And there are anti-parallel strands in this DNA. That is, if your carbon, this 1, 2, 3, 4, 5, on the fifth carbon there will be always a phosphate group. Yeah, and that will go into this chain 5 prime to 3 prime end. On other side it goes like this 5 prime to 3 prime end. So in the center is your nitrogenous base pairs. The adenine, they are making a di double hydrogen bond, and guanine and cytosine, they are making a triple hydrogen bond. and yeah, that's why it's anti-parallel and they run opposite to each other. And further, Watson and Crick model. So the adjacent deoxyribonucleotides, they're joined in chain by a phosphodiester bond. So they are, there's one bond here, there's one bond here. So this phosphate group is making two ester bonds. So that's why this term is known as phosphodiester bond. And they're making 5' end of one carbon here and 3' carbon there. So the two chains are helically around with each other with sugar and phosphate bond outside and base inside. Hydrogen bonds base with each other. It's an anti-parallel strand, and coiling is a right-handed side. We will know what is right-handed side. It mainly means it moves in the right-hand direction. And one complete turns includes 34 angstrom. That means it has major and minor groove, and this major and minor groove together, it is a size of 34 angstrom. And within this 34 angstrom, there are 10 nucleotides present. 10 mononucleotides are present. This side is of 34 and it includes your both major groove and minor groove. So now let's discuss what is major and minor groove. Major groove is basically something when it's bending towards from the central axis. There's a deep and thick gap from the central axis. That is known as your major groove, and the size of around 2.2 nanometer. Whereas the minor groove, it's of 1.2 nanometer only. Yeah. The height of this is around 1.2 nanometer, and the height of major groove is around 2.2 nanometer. And together they make the length of one turn to be around 3.4 nanometer, and within this one turn there are around 10 nucleotides. How many? There are around 10 nucleotides, and the diameter is most commonly is around 2 nanometer. And this DNA that we are seeing here is the dimensions of bDNA, which is the most common DNA that are found in our body. This is your bDNA, right? This is 3' end, this is 5' end. This is the major groove. Here's the minor groove. This is a space filling model. This is a ribbon model, and so on. A, G, C, T. Yeah. So that's the basic concept behind this. And there's also a thing called rise. The distance between two nitrogenous base pair is known as rise. And sometimes there is an angle between two nitrogenous base pair, like they could be tilted, like one nitrogenous base pair is this, one is this. So angle between them is known to be as tilt. Okay, because based on these dimensions we will get to know about three types of DNA, that is aDNA, bDNA and zDNA. So we will get to know about three types of DNA, that is aDNA, bDNA and zDNA. Okay. I hope everything is fine at your end. Lecture is going fine, my voice is fine. Okay, so we have discussed the basic structure of DNA so far. Now let's take it to the different types of DNA, how various types of DNA looks like. So this is the summary of what we have discussed so far. The features of DNA double helix. They are coiled together from right-handed helix. They run from one parallel 5' to 3' end, and orientation is opposite. And nitrogen bases of opposite chains pair to one another to form hydrogen bonds. And base pairs stacks on each other located inside the helix. And each turn completes around 3.4 nanometer and contains 10 bases pair. And each turn along the molecule contain one major and one minor groove. Yeah, it contains one major and one minor groove. So there are three types of DNA, that is aDNA, bDNA and zDNA. So the aDNA is right-handed double helix structure, which is compact with 11 base pair per turn and 2.3 nanometer in diameter. And the bDNA, so we can see here it has 10 base pair per turn, and here it has 11 base pair turn. So it has one more base pair, so that's why they are more elongated. And the diameter is also broader, around 2.3 nanometer, and here the diameter is 2 nanometer. So in the diameter is also it's bigger, broader, and this is intermediate. This is intermediate in broader and intermediate in the height. Whereas the zDNA, it has 12 base pairs, is the highest elongated, but the diameter is the least, so it's the thinnest in diameter, 1.8 nanometer. And it's a left-handed, not the right-handed, and it's the least common find out DNA in the human body. Yeah, and it's give a zigzag structure. It's not like helical structures in A and B, but this give a zigzag structure. So like this. So these are all right-handed helix, A and B, and this is zDNA, this is left-handed helix. This diameter is less here, the least. This is the intermediate, this is the highest. Yeah, and this is the most tallest because it has 12 nucleotide base pairs present in the zDNA. This has 10. Yeah, this has 10. This has 12, and this has 11. aDNA. Okay, that's why there is a difference in these structures. And this give a Z type of structure, and they give a helical structure. Moreover, some terms. So as we have discussed in the bDNA, major groove is bigger, thicker and broader, and the minor groove is smaller here. But in the aDNA, it's the opposite. Minor groove is more thicker and broader, major groove is smaller. And then there is a rise, that is the distance between your two nucleotides, and there is a tilt, that is the difference between the angle between your two nucleotides. Yeah, so these terms you should remember, because based on that your aDNA, bDNA and zDNA, they are highly differentiated from each other. Okay, so this aDNA, if we see from the proportion of overall proportion, this is short and broad, this is longer and thinner, this is elongated and slim. So why is it short and broad? Because the rise per base pair, so the distance between two nucleotides, that is rise per base pair, this is 2.3, this is 3.32, and this is 3.8. So distance between two nucleotides, it has the highest distance here, 3.8. This is intermediate, 3.32, and this is 2.3. So that's why it's the shortest in height. This is the longest, and this is the elongated. And the diameter, if we see, this is 25, 23.7, this is 18.4. So this is slim, thinner, broader. So these terms explains the overall proportion. Then comes the helix rotation. So this is right-handed, this is right-handed, and this is left-handed. Yeah, right-handed, right-handed, and left-handed. And the base pairs per helix repeats are one base pair per helix, one base per helix, and here are two base pair helix. How many in per ton? How many nitrogenous base pairs are? Here are 11. This will be 10, and this will be 12. That's why it's the elongated. This is longer, and this is shorter. Then comes your third term, that is base pair tilt from the perpendicular. This has the highest angle, plus 19. This makes it minus 1, minus 1, 2, and this is minus 9 degrees Celsius. Yeah. Helix axis, so major groove. So this is the major part in this, that is your major groove. The major groove here is extremely narrow but very deep. And this is wide intermediate depth. This is flattened out the helix on the surface. Whereas the minor groove here, this is very broad but shallow, narrow with intermediate breadth, and this is extremely narrow but very deep. So that's the major difference between your A DNA, B DNA, and Z DNA. Cool. Some more diagrams. So there's also further classifications of DNA, that is, they could be a linear DNA, circular DNA. The linear DNA are the one which both ends are free. Yeah, they are both ends are free here, like this. And the circular will not have, it will be like, it will have no ends free in the prokaryotes. And they are mostly present in the eukaryotic cells, linear, and circular are found in the prokaryotic cells, and so on. So this is an example of the same. So now it's a Q&A time. So students, answer the first 5 questions, and then we go to the next 5 questions. Yeah. So DNA stands for D-ribo-acids. What is the shape of DNA? Who established the structure of DNA? Adenine always pairs with, sides of DNA ladder are deoxyribose, and guanine always pairs with. So sorry, till 5. You have to answer till 5 questions. These one. So answer them quickly in the chat box, and then we continue further, students. So answer them at once, bachche. Answer all of them at once. Answer like 1, 2, 3, 4, 5. Yeah, all are correct. Shabash. Now answer the next 5 questions. Guanine always pairs with. What is the complementary sequence towards this? And the two signs of DNA are held together by which bond? And DNA are composed of repeating subunit of what? And what are the 4 bases that make up the rungs of DNA ladder? So answer the next 5 questions and answer them like 1, 2, 3, 4, 5 together, bache. Like now 6, 7, 8, 9. So in your chat box, just answer these as 6, 7, 8, 9 by one person only. So in this way, it's very hard to find out the answers coming up in the chat box variedly. So I will bring it to that. So just answer 6 to 10. Nice, nice. 10th number answer. It's adenine, thymine, cytosine, and guanine. All the 4 nitrogenous base pair. Is the nucleotides, but not nucleosides. Yeah, Johnson gave all the answers correct. On the 9th, it will be nucleotides. And the 7th answer is, yeah, just write down the complementary sequence. That will be T, T, A, C, G, T. So this will be DNA is composed of nucleotides, hydrogen bonds, guanine pair with a cytosine. So that's it. So we have understood the structure and functions of DNA as a genetic material, and we can correlate with the various downstream topics of replication, transcription, and translation now in our upcoming lectures. So that was a reference. So whatever the presentation I will be sharing with you, so I will share that material with you also side by side. So if you want to know, I have some books also, you can use them. This is a technique book by Wilson and Walker, a very useful book. Some of other biochemistry books might be helpful for you. This is Lehinger, Harper, and Lippincott. Yeah. The Genetics and Molecular Biology book. Concepts of Molecular Biology. So this will take some couple of minutes in order to upload. Okay, so here students, we can take 5 to, it's 12:15 now, so we can take we can meet around 12:20 or 12:23. I just need some energy to revive myself, and then we see us again. We continue with now various types of RNA transcription and translation. Okay, class, let's start the next session. Bache, try to open in, are we opening in the mobile? Try to open in the laptop, and you should have the latest PPT software. I think for all others it might be opening. Okay. So usually I start my lecture with structure of DNA, then I comes to the molecular biology part. So what is molecular biology? It's basically study of molecular underpinning of the process of your replication, transcription and translation of the genetic material. So it's actually including your animal cells, plants, microorganisms, molecules, populations. They're all coming together and they are being fixed, being part of it. So much of the work in the field of molecular biology, it's quantitative, and recently has been done at the interface of molecular biology and computer science, that is your bioinformatics and computational biology. So in this, the key player role that are playing in is our DNA, RNA and protein. So everything is revolving around them. That is DNA, RNA and protein. And gene is acting as a hereditary unit with the DNA segments that carry the genetic information. They are called genes, and it's normally a stretch of DNA that codes for type of protein or for the RNA chain that has function in the organisms. So genes hold the information to build and maintain organism cells and pass genetic traits to offsprings. So this chromosome has gene 1 and gene 2, and on the upper figure we can see there is a DNA revolving around the histones and then around the sister chromatids. And these chromosomes, they come into the 23 pairs of chromosomes present in the nucleus as a cell. So DNA this we have discussed in detail just now. Everything has been discussed. So now comes RNA. So RNA, it's a biological important type of molecule which consists of, again, your long chain of nucleotide units, and each nucleotide consists of nitrogenous base pairs, ribose sugar and a phosphate group. So all these things are present in your RNA, and you can see there's a single chain, and then there's a double helix, and then again single chain, then again double helix, then again single chain. And the side backbone is a sugar and phosphate, and in the top here is the nitrogenous base pairs. And you have your double-stranded RNAs from the genetic material of some of the viruses. We can see in this genetic material being present of your RNA, double-stranded RNAs. And further, there are three different types of RNAs, that is messenger RNA, ribosomal RNA and transfer RNA. The messenger RNA, they codes for protein. The ribosomal RNA, they are known for translation, and transfer RNA also for translation. All these organisms. And then the post transcriptional modification, small nuclear RNAs are there, snRNAs and yRNA and telomerase RNA. and beyond that the regulatory RNAs, that is antisense RNAs. But our main task is to understand mRNA, ribosomal RNA and tRNA. So messenger RNA, it's act like a postman which take information from DNA and give it to the ribosomal RNA and your tRNA outside the nucleus in the cytoplasm. So from there your specific amino acids group will be producing and things will go on. So here we can see there is a coding strand and there is non-coding strand, and the corresponding the coding strand you will start having the production of a messenger RNA with G, U, U, A, C, C, G, A, U like this. So the mRNA further is then exported from the nucleus to the cytoplasm, where it is bound to the ribosomes and translated into its corresponding proteins formed with the help of transfer RNA. So in prokaryotic cells which do not have nucleus and cytoplasm compartments, so mRNA can bind to the ribosomes while it is being transcribed from the DNA. Okay. So this statement, it's saying mRNA is then exported from nucleus to cytoplasm, where it is bind to the ribosomes. Okay, your mRNA is done, it's get moved out of the nucleus and it goes to the cytoplasm, and then from there it is bound to the ribosomes and then translated to its corresponding protein from with the help of tRNA. So this statement itself says a lot of many things. So I will take you to the some animations now and will explain you about transcription and translation, how mRNA moves out of there and start acting like that. Yeah, so let's start with that.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order, but small differences in the sequence of the bases in our genes make each person unique.
So that's why we are different from each other, just because of the differences in the ATCG. Who is this? Chandrima Ghosh. Please contact with Vatsal sir or please give my reference. So this was about DNA. Now let's talk about protein.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
Yeah. So let's see how different same gene present in different organism works. It's like you have your jellyfish. It has a fluorescent genes. We can put them in the fish. We are having these spiders. They are producing silk web. We can produce them in the yeast to produce them. So we can use the same gene in different organism to get our work done.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
Yeah, very nicely explained what we have talked since morning in a nutshell. Okay. Now, transcription and translation.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life. Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So students, tell me in the chat box. So we are producing mRNA now, not DNA. So what will be the corresponding sequence to it? If it is GAs, what will be the corresponding sequence to it? In the case of RNA, remember that A in DNA pairs with U in RNA. Remember this thing during the transcription, A pairs with U in RNA. So G will pair with it's going a bit far, it's lacking. So tell me students, it's C. A pairs with U, C pairs with G, U, and so on.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So tell me students, AGU, it will correspond to which one? Will it be serine, phenylalanine, valine, or valine? will be serine. Then UGC, think about it. ACG, cysteine, and so on.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
I hope this transcription and translation you will never forget in your life. This will be remain with you. Okay. So this is the lecture that we were going through, basics of molecular biology. So far, any questions, students? Any questions so far, student, till here? Only then we will continue further. What is Anjum? What one more, please? What is the thing we want to discuss? You mean transcription and translation? Okay, let me know what you mean. We will continue with the transcription and translation now, actually. So the transfer RNA, as we know, it's a clover leaf model in which on the top is your amino acids are attached with the help of ester bonds, and correspondingly mRNA will attach to the tRNA with the anticodons here on the top, G, 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 C, C, C. So it's a small RNA chain of about 80 nucleotides that transfer the specific amino acids to a growing polypeptide chain at the ribosomal site of protein synthesis during translation. So ribosomal RNA, it has 50S and 30S subunits with 70S, and 60S and 40S subunit with 80S together. So there are two types of ribosomal RNA, and the main difference between RNA and DNA is they lack hydroxyl group at the second carbon. So they are ribose and deoxyribose, and they're single-stranded and double-stranded. Whereas the proteins are known as polypeptides, they're made up of amino acids arranged in a linear chain and folded into globular form. And the sequence of amino acids in a protein is defined by the sequence of genes which are encoded in the genetic code. And genetic codes are specifically around 20 amino acids in them. Yeah, there are around 20 amino acids. So here we can see lysine, valine, phenylalanine, glycine, like so on. And the cysteines are making disulfide bridges and so on, and it's making a very nice structure. And they goes into the quaternary structure, and we get them from the pulses, peanuts, red kidney beans, fishes, white chickpea, red meat, eggs, chicken breast. So all are the sources of your proteins at the end. So the basic players that are playing in your molecular biologies are DNA, RNA, and protein itself. So DNA could self-replicate, self-replication could happen. Then the transcription will happen, which will lead to the RNA, and that RNA will lead to the translation and leads to the protein produce. So these are the basics, which are the and this whole process is known as central dogma of life. So I will now do the last topic of DNA replication, then the transcription part, we do it tomorrow. So in the DNA replication, which is a biological inheritance, which is semi-conservative nature, which goes from parents to the offsprings, they give the 50% of their genes to their offsprings. So in that, what is happening is majorly your topoisomerase, your helicase, your single-stranded binding proteins, SSBs, your DNA polymerase. Yeah, so all these enzymes are playing big role. So what is happening is there's a strand called 5' end and there's a 3' end. So this is 5' end, which is stopping at 3' end here, and this is 3' end, which is stopping at 5' end here, right? So what will happen? Your DNA replication is always happening in the direction of 5' to 3' end. So if it is starting from here 5' to 3' end, your DNA polymerase will start producing it. But initially, first that topoisomerase will unwind your the double helix structure, and the helicase enzyme will break the bonds between the two nitrogenous base pairs. And then SSB proteins, they make stabilization of nucleotides, and then DNA polymerase helps to produce your corresponding nucleotide base pairs. So in this way, your first strand will be your leading strand. This will be your leading strand. On the other side, the lagging strand, it will also move from in this direction, and this is going in this direction. This in the leading strand, it's going in this direction, 5' to 3' end, and the lagging strand on the top, it's going in the opposite direction, where it will keep producing these Okazaki fragments. So they are not happening in the continuous manner. The Okazaki was a great scientist who discovered these fragments, so named on the Japanese scientist. And these fragments are filled with the help of DNA ligase. And your DNA polymerase here also doing his job. And RNA primer and primase, they helps to initiate that direction of nucleotide base pairs. So in this way, all primers, all enzymes, they're playing a big role in this DNA replication in order to have a successful replication at the end. And we can see that it's made a shape like, it's also called replication fork. So it's made a fork like this, kanta, that we use for eating food and so on. So that's it, students, for today. The transcription, we will do it tomorrow. Please let me know as I will Jigmet and Biswajit. We will do about transcription and translation in our upcoming lectures. So I want to start with transcription here only, how the transcription process is done, then the translation process. So I will discuss it in the next lectures. Otherwise, we will not have much topics to do so. Cool. How much time does it take to synthesize a protein in the cell? Oh, that's a good question. Maybe in milliseconds. I have to Google that. That's a never thought of. 20 seconds. Yeah, I was thinking right. 20 seconds to several minutes. It depends upon which kind of proteins have. Yeah, it depends. Anjum, which slide you didn't get? The transcription and translation. Okay, I will explain it. No worries. I mean, you can watch that video also once more time. Yeah. Sorry. Extremely sorry. So basically, in transcription, it's a process of creating a corresponding equal amount of RNA of your DNA. So RNA is acting like a copy which moves from nucleus towards cytoplasm. If the DNA is producing RNA, it will be known as transcription. But if RNA is producing DNA, that will be known as reverse transcription. So basically, during the transcription, your DNA sequence is read by the RNA polymerase, so which produces an anti-parallel RNA strand. And in this, instead of thymine, uracil is included. So during the stretch of DNA transcription, so RNA molecule is called transcriptional unit and encodes at least one gene. And this will encode further for a protein, results in the transcription in the messenger RNA. So let's see here. So DNA that is reading from 3' to 5' end during the transcription. So this is your DNA. So it's got unwinded up. Yeah. So it's 3' to 5' end. And on the other hand, the 5' to 3' end on the corresponding, your RNA polymerase will add it. And this RNA polymerase will produce a complementary mRNA sequence. Like A, then T will produce. And this will get removed and become a mRNA. It will have also hairpin loop and so on. Yeah. And then your RNA polymerase will get removed. So remember, it could produce this DNA replication in this way, but for the mRNA, it will not have T, it will have all uracil. And also during the RNA transcription, our DNA has exons and introns. Exons are the coding regions and introns are the non-coding regions. So exons will remain there and the introns will be kicked out. And because of that, you will have exon 1, 2 and 3 RNA splicing. So RNA synthesis and processing will happen. And then reverse transcription. In the reverse transcribing viruses, replicate the genomes by reverse transcribing DNA copies from the RNA. So this is reverse transcription which is happening mostly in the viruses. So if HIV RNA is there, it will produce a corresponding HIV corresponding DNA with the help of your reverse transcriptase enzyme. So in total, what we can see is in the general cases, DNA could self-replicate, and after DNA with the help of transcription produce an RNA, and then RNA could produce protein. Okay, but in the special cases, RNA with the help of reverse transcription reverse transcriptase enzyme could produce DNA, and RNA could also self-replicate. That's a special case. But it will never happen that DNA will go and start to produce proteins. This will never happen. And proteins goes to DNA. It will never happen. Yeah, so these two things will never ever happen. Or protein goes to RNA back. So these are exceptional cases. So basically in translation, what is happening is the first stage of protein biosynthesis. So in this, mRNA produced by transcription is decoded by the ribosome. So you will have your ribosomes will come to the mRNA and they start to decode. So here we can see the mRNA that came in that figure in the diagram. They got this tRNA got attached to the alanine here. Yeah. Then this alanine, valine, leucine, glycine, methionine, they're all working like that. and this ribosomes got attached to this tRNA, and then glycine will attach to it. And then further, the amino acid, the alanine is the the corresponding mRNA codon, and the corresponding tRNA will come with the addition of your amino acids on top, and this will attach further, making a long chain of a protein amino acids, right? And then other glycine will come and will attach to it, and then glycine will attach to the alanine, and then phenylalanine will come, and so on. So this way we have our complete chain of our amino acids with the help of your ribosomes, yeah, tRNA and mRNA. So they are all coming together to play this role as of a translation process. So I think this one is clear. I hope this one is clear from the all perspective now. The transcription, translation, DNA replication, DNA, RNA, various types of DNA we have discussed. We have discussed various type of RNA, and yeah. So how the protein increase in the case of a uric acid patient? It's something I so far remember. Tryptophan. There is some mismanagement of enzymes are happening. I do not remember the exact reason behind it. It's in the medicinal biochemistry we are doing it. I will get the answers later, but at the moment I'm not aware of it. Sorry. Okay. Anyone else? Any further questions? I will wait. Yes, Miss Vijita. Okay, if no, then we are done for today's workshop. We see us tomorrow at 11 a.m. till 1 p.m. then. Until then, thank you very much for today's class, being part of the class today. And we see us tomorrow again, then same time, same place. Okay, go through all these books that I have shared with you today. You will get plenty of materials from me. So this is what I do always whenever some students are joining me. I try to provide as much things I have so that could be useful for everyone. Any problem, student, let me know. I will help you, yeah, in that. Okay, students, thank you very much. Bye bye. Take care.